Hello everybody. In this video we will be talking about the cell junctions that hold cells together and perform a bunch of different roles in tissues. And we'll close out with just a review of the definitions of basal and apical ends for epithelial cells, because that came up in the Edpuzzle videos a little bit as well, and it all kind of ties together into one story. So we really have 5 different cell junctions that we're responsible for, and 5 different junctions that you'll have to review in your e-report. And they break down into tight junctions, adherence junctions, desmosomes, gap junctions, and hemidesmosomes. And we'll review them one by one, both their structure and their function, because those two go hand in hand and they're quite important. So for tight junctions, here is one cell, and here is another. And in this example, these would be epithelial cells, so cells that either line our skin, so how our body faces the outer world, or cells that line our intestines, our guts. And so that's still facing the outer world, but it's the outer world that we bring into our body, right? Where this tube within a tube notion that kind of runs through all of biology. We bring the outside world into us so that we can extract nutrition and all of those things from what we consume. In both of those cases, we need a watertight seal. We need there to be no room between cells that water or any solutes can get through. We need everything that comes into us or goes out of us to go through the cells. That's really important because as far as our skin goes, if water and other things could just kind of seep into our bodies between our skin, we would be susceptible to a lot more contamination. We would swell and explode in a pool, and we certainly don't want that to happen. And for the gut lining, in order to absorb nutrients from what we digest, we need those nutrients to be absorbed exclusively by cells. And if some of the breakdown products and metabolites of digestion from the small intestine were able to leak between cells, they would never be absorbed, and we would never be able to use those molecules. So there's a good reason for there to need to be a watertight seal between epithelial cells. For tight junctions, the way that that is achieved is by 2 different proteins that are expressed on the surfaces of neighboring cells, of all cells, and those 2 proteins interact with one another in a zipper-like fashion and create this watertight seal. So now this intermediate region between these 2 cells is completely blocked from anything leaking in between them. The 2 proteins that are responsible for this zipping up, for this exclusion of water, come down to claudins and occludins. Occludins like occlude, or to block. Occludins. And as you can see in my diagram, both cells that are adhering to one another express claudins and occludins. The claudins and occludins interact with one another, but they're expressed in an alternating pattern. So here we have an occludin on the left-hand cell and a claudin on the right, and here we have the claudin on the left-hand cell and the occludin on the right. But the most important thing here is that these proteins zip up with each other. They interact tightly with each other, basically zipping these 2 cells together in a watertight way. So that's what we want to know about tight junctions. Now, adherence junctions and desmosomes, I'm going to talk about simultaneously, side by side, because they share a lot in common, which makes them easier to remember because you don't have to memorize 2 things separately. But they're also somewhat easily confused as well because they have so much in common. So we'll do our adherence junctions on the left, and we'll do our desmosomes on the right. So let's first talk about how they are similar. Well, they are similar in that both of them are going to culminate on this transmembrane protein called cadherin. Cadherin. C-A-D-H-E-R-I-N. So they have that in common, desmosomes and adherence junctions. And cadherin is called cadherin because that's meant to remind us that these proteins are adhesive. They adhere to one another through calcium. That's the cad part, the Ca. So cadherins adhere through calcium. So there's a calcium ion that links these cadherin proteins together. The reason why this is a stable interaction right here and right here is because a single calcium ion is bridging these 2 transmembrane cadherin proteins that are expressed on these adjacent cells. So they both have that in common. In adherence junctions, here on the left, The cytoskeletal component that is linked directly to these cadherins, well, let me do that a little differently actually and be even more accurate. The cytoskeleton component that is linked to these cadherins is actin. And the way that I remember that is A for adherens, A for actin. So adherence junctions use actin in their cytoskeletal component. And what we're essentially doing is we're tying the cell cortex of one cell to the cell cortex of another. So the cell cortex of the first cell is made up of actin. That cell cortex is linked directly to cadherin. Cadherin is interacting with calcium. Calcium is bridging to the other cadherin. And that other cadherin is attached to the cell cortex of the neighboring cell. So these two cell cortices are interacting with one another indirectly through cadherin and this calcium bridge. For desmosomes, the cytoskeletal component that we are linked to is intermediate filaments. And to be a little bit more specific, it is actually the keratin intermediate filament that most cells share. So now keratin is linked to the keratin of one cell on the left is linked to the cadherin protein, which is bridged to the next cadherin through a calcium ion, and that next cadherin is interacting with its own intermediate filament network in the neighboring cell. So this cell junction ties the intermediate filaments of neighboring cells together. Now, functionally, why might we want these differences? Well, if keratin and intermediate filaments in general are for tensile strength, it's to allow the cell to withstand the stresses of stretching, then this desmosome is now allowing tissues to combat the effects of stretching. As we stretch and deform and put these tissues under tensile strength, within each cell, the intermediate filaments are absorbing that stress, but now the intermediate filaments of adjacent cells are connected to one another through hemidesmosomes, so the entire tissue can stretch without tearing. If we think of the role of actin being largely in cell movement, external cell movement, then we realize that by tying cells together by adherence junctions, we're actually allowing moving cells to flex and move with each other. They can influence each other's movement. As the cell cortex of one cell deforms, it is bringing and heaving another cell with it. So there are different uses for these two junctions. So to summarize this component, we have adherence junctions, which is the tying of actin from one cell to a cadherin transmembrane protein, bridged to the cadherin of an adjacent cell through a calcium ion, and of course that adjacent cell's cadherin is also bound to that cell's actin. For desmosomes, it's the intermediate filaments of one cell linked to cadherin. That cadherin is interacting with the cadherin of a neighboring cell through a calcium ion, and that neighboring cell's cadherin is linked to its intermediate filaments, providing this coping with tensile strength. So now we can move on to hemidesmosomes, because hemidesmosomes are going to allow us to leverage some of what we just talked about with desmosomes. So let's talk about what's in common there. So for a hemidesmosome, we start with a single cell, and that desmosome similarity is meant to remind us that we are going to again be talking about intermediate filaments. Again, we're going to be talking about keratin, specifically the IF keratin. So this is once again a tensile strength connection. But hemi means half. So here the cell is going to only be half connected. As opposed to being connected to other cells on either side, we're only going to have a half a connection, and in fact that half a connection is not going to be to another cell at all, it's going to be to the extracellular matrix. What do we know adheres to the extracellular matrix? I heard someone say it, integrins, and so that's what we're going to use as our transmembrane protein here. So in a hemidesmosome, we have the intermediate filament component of a cell's cytoskeleton tied directly to an integrin. That integrin is a transmembrane protein, and it is doing what integrins always do. It's interacting with the extracellular matrix. If this extracellular matrix is collagen, then we need a fibronectin protein right here, as we talked about in another video this week. Instead, most of the time, if this is an epithelial cell, what we are actually adhering to is a very unique component of the extracellular matrix called the basal lamina. And that would make these lamins, and the integrins are binding directly to these lamin proteins of the basal lamina. So if we think about your skin, what we have is bone, of course, and then muscle on top of the bone. And then we have this basal lamina surface layer made up of this protein meshwork, this very specific ECM. And then we have your dermis, your cells linked to the basal lamina. That's why as you I'm doing it right now, if you put your finger on your hand and you push your skin around, it moves a bit, but it doesn't slide all over. In other words, as skeevy as it is to say, you can't grab the top of your hand and pull it all the way around to the bottom of your hand as though it was some kind of glove. The reason for that is your dermis, the cells of your skin, are physically connected to the underlying protein meshwork of your body. That's the basal lamina, and now you know how that connection is mediated. It's mediated through the intermediate filaments of your skin cells bound to an integrin transmembrane protein, which is then adhering to that basal lamina. So that is a hemidesmosome in that example. We'll take a quick detour here as well and define these terms that were bothering you just a little bit in the lecture. This epithelial cell now has polarity. Polarity in the sense that this is the outward-facing side of the cell. So in other words, if this is a skin cell, then here is where the air touches the skin. If this is a gut lining cell where we bring the outside world into our body, then here is where we are in contact with digested materials in the small intestine, in the stomach, and in the large intestine. That makes this side the inward-facing side. This is the side that is facing the musculature. This is what's connected to the rest of us, the inward-facing side. And since this cell has polarity, it has two different ends, we give those two ends a different name. Any outward-facing side of a cell is called its apical end. So if you rub your hand on your skin right now, you're touching all the apical surfaces of all those skin cells. The parts of your small intestine that have the villi that are absorbing the nutrients from your digested food, those are the apical ends of those gut lining cells. The part of the cell that is adhered to the basal lamina, well, this is an easy one to remember. We call that the basal end. So the anchored end, or the bottom of the cell, if you want to think of it that way, is the basal end because that's the end that's adhered to the basal lamina. That's where the integrins are involved, and that's where you find your hemidesmosomes. So great. Trucking right along now, we've only got one left, and that is our gap junctions. Many students find gap junctions the easiest to remember because they're the least complex, and they just make sense. Cells need to be able to talk to their neighbors and tell their neighbors what's going on. Now, of course, cells talk to each other by exchanging molecules. Second messengers, hormones, steroid hormones, even ions can be used to talk to neighboring cells. So if cells are going to have this ability to communicate with each other adjacently, and they have to have this ability to pass molecules onto each other, then really, if we leverage what we've learned about in previous lectures, we just need there to be a channel here. An open portal from this cell that allows things to move in and out. And then ideally, if we want these cells to be able to interact in real time, we should have a complementary channel right here. And now molecules can move through this channel and the adjacent channel and enter the cell on the right. And molecules can move through these channels in the other direction and enter the cell that's on the left. And that's all a gap junction is. You can think of a gap junction as two adjacent channels in two adjacent cells that connect to one another and then create an open portal between those cells for small molecules to move through. These channels are distinct and unique, though. They're not just any channel, and so they get a name, and these are your connexons. So connexons are these half channels between adjacent cells that connect to one another and allow molecules to move freely between those two cells. Not all adjacent cells have connexons, because we don't want cells necessarily talking to each other in all tissue types. But the examples that I give in the lecture are certainly appropriate. So involuntary muscles, such as cardiac muscle, the way that we have a wave of contraction from one cardiac cell to the next, to the next, to the next, is because the ions that are causing that contraction are moving through these cells from one to the next through connexons of gap junctions. And so those are the 5 cellular junctions that we have to talk about. We've got tight junctions, which are watertight seals between epithelial cells. Claudins and occludins expressed on each cell zip up to create this watertight barrier. Adherence junctions and desmosomes are involved in absorbing different kinds of stress and movement. Adherens, we have A for adherens, we have A for actin. So the actin of one cell connects to a cadherin, and that C in cadherin reminds us that we need a calcium ion bridge to the next cadherin of the neighboring cell, and that cadherin is tied to actin as well. So it is actin, cadherin, calcium, cadherin, actin to connect 2 cells together. Desmosomes rely on intermediate filaments. So here it is keratin to cadherin, to calcium ion, to cadherin, to keratin. This allows tissues to be stretched without tearing. We've got hemidesmosomes, where we stay in the realm of intermediate filaments, but here the keratin is tied to integrin molecules, integrin proteins, and those integrin proteins are not grabbing onto an adjacent cell, they're grabbing onto the basal lamina. This is how epithelial cells remain adhered to the underlying matrix or the underlying ECM of our musculature. And finally, gap junctions are open portals between cells mediated by connexons, where cells can transmit and transfer small molecules between each other. So that's it for cell junctions, and we even defined basal and apical for you. Hope it made some sense.